Hi guys, so today we will be starting a topic from the 11th boards. It's biomolecules, right? It's a very important chapter. It comes both under chemistry and biology. In biology, we will deal with mostly the biological aspects of these molecules, and in chemistry, you will deal with the obviously the equations and how they form the bonds and all of that. So now starting with biomolecules, very briefly, very easily if we break down the word, bio is life and molecule is molecule, right? Any chemical substance. So all the chemical substances that would constitute life, that would make up what we know as life, that is essential for life, would become your biological molecule or biomolecule. Now biomolecules inside the cell, so all the molecules in the cell will form what we know as the cellular pool. That includes whatever constitutes your cell wall, the cell membrane, the cytoplasm, the organelles, and whatever your organelles are also making. If we want to classify the cellular pool on the basis of their state, so if they are in an aqueous state, dissolved in water or colloidal, they, on the basis of that we can classify, or we can classify them on the basis of size. So those that are in a dissolved or colloidal state are your aqueous molecules. So, for example, every protein, let's say, which is in the cytoplasm, which is in a certain form of a colloid, or your certain hormones, they are all in a colloidal state, right? And non-aqueous states of the cytoskeleton, the cell membrane, the organelle structure, these are in a non-aqueous or a non-dissolved state, right? Closer to solid state. Now, coming to by size, if we were to classify, then we classify them on the basis of Daltons. Dalton is the size unit which we use to measure molecules, macro and micromolecules in biology as well as in chemistry. Now, here any molecule which is within 18 to 800 Daltons would be called the biomicromolecules, and anything above 800 Daltons would be biomacromolecules. Sometimes in some books you would see this number to be 1000, so it varies, but yeah, roughly 800 to 1000 Daltons. Anything larger than that would be your biomacros, and anything smaller would be biomicros. Now, how do we figure out what our biomicros and biomacros are? There are two experiments, very interesting, easy experiments that we can very easily do. One of them will figure out the organic matter, and one of them will figure out the inorganic matter in any given tissue, let's say. So let's start with the inorganic molecule. You can take any tissue, you can take, let's say, a part of an onion, right? Any vegetable, a piece of a liver. It's very easily available, and you will have to dehydrate it. So let's say the tissue that you take originally weighs about 100 grams, and then you pop it in the microwave and you dehydrate it, or you dry roast it, all the water evaporates. So when you dehydrate something, you basically lose all the water, and when you have dehydrated it, you weigh the dry tissue again. So now your dry tissue, let's say, weighs 20 grams. From this, you can very easily say that the water content was about 80 grams, correct? Now you take this 20-gram dry tissue and you burn it. When you burn something, obviously the carbon dioxide and whatever water is left will also escape. So carbon dioxide and water in the form of vapor will escape. Whatever then will be left would be the non-carbon containing compounds. They are your inorganic compounds. So what you would get is basically the ash. When you will weigh the ash, let's say it turns out to be 1 gram in weight. So then you can say that mostly there was about what? 19 grams of organic molecule, and whatever you're left with is your organic matter. Correct? So that's a very easy way to figure out how much of water and organic and inorganic compounds a particular tissue consists of. Now, if you would analyze this inorganic matter, right, using chemical methods, you would figure out what kind of substances are in it. So you will find calcium, you'll find phosphorus, you'll find a lot of chlorine, stuff like that. Correct? So that's how we figure out the inorganic components. Coming to the organic matter that we have. So if you take a wet tissue and you grind it in trichloroacetic acid, right, you will get a solution. Now you will filter this. Okay, it's a mixture actually. You will filter it. When you filter it, some of the substance will obviously fall down as the filtrate, which is the acid soluble part, and some would stay stuck to the filter paper, which is your retinate, which is the acid insoluble pool. Upon measuring the molecular weights, it has been seen that the biomicromolecules come in the acid soluble pool and the biomacromolecules come in the acid insoluble pools. In this biomacromolecular pool, we have found four very interesting structures. Why interesting? Because along with being macromolecules, all the inorganic substance was found in the retinate, almost all, and all the organic substances were found in the filtrate. However, four organic molecules, the polysaccharides, the lipids, the proteins, and the polynucleotides, which are all organic molecules by the way, were found in the retinate in the acid insoluble pool. Why? Because they are larger, because all of them are larger in size than 1000 Daltons except lipids. Then the question arises, why did these lipids not get dissolved and get into the filtrate? That happened for a very particular reason. So when you're taking any tissue, where is the lipid in that tissue? The lipid is obviously in free state, but it is mostly also present in the cell membrane, in particular organelle membranes, correct? Because it makes up the structure. So when you have ground it, the lipid immediately breaks down into smaller particles and forms vesicles. These vesicles are insoluble in acid, and therefore all your lipids will then be retained in the retinate in the insoluble pool. That is why we find lipids in the insoluble pool and not in the soluble pool, even though it is smaller in size, much smaller in size. I hope this was clear. The questions that you get are questions like why are lipids in the insoluble pool? What do you find in the insoluble pool and the soluble pool? These experiments and the definitions, obviously. We would further do carbohydrates, proteins, fats, and nucleotides in details. I hope you enjoyed the video. Thank you.